Hi everyone, this is Zhongtao Hu, a postdoc from Washington University in St. Louis. Thank you for your attending my presentation. I'm going to introduce a tunable focused ultrasound device for high-precision drug delivery to the mouse brain. Focused ultrasound-induced blood-brain barrier disruption is a promising tactic for both clinical and preclinical applications. MRI-guided fast devices are the most commonly used system for fast BVD. Preclinical systems also included ultrasound imaging guided system. However, the devices for preclinical research are expensive, bulky, and with a high tactic barrier. Moreover, the focus region of the commonly used fast transducer are too large that often cover the whole brain region, which limited the spatial precision in preclinical application. As shown by the two CE MRI and Evans blue imaging, the elongated fast beam induced BVD across almost the whole brain region. The object of this study is to develop a tunable, easy-to-use, and high-precision fast device for fast BVD in mice. Our design fast device combines in-house midi-fast transducer with a commercially available stereotactic free. The key hardware component was a midi-fast transducer, and the transducer are made of single-element concave-shaped PZT with a potential of only 30 mm, which is smaller than the commonly used fast transducer. 3D printed adapter was used to connect the fast part with the stereotactic frame through the shaft as shown here. The transducer was easily attached to the adapter using the two magnets. Each PZT element only cost $80, and all other parts are 3D printed. The frequency of the fast transducer can easily count by changing the fast element, making this device tunable. Here is the picture of 3 midi-fast transducers are made with different frequencies for the goal to access whether higher frequency can achieve more precise BVD opening. We use established and easy-to-use stereotactic procedure to move the fast transducer for accurate brain targeting in the following two steps. In step 1 is to target the bregma. A dot was drawn on the mouse scope to indicate the location of bregma, which was visible through the scope, and a pointer was placed in the holder, and its position was adjusted by the stereotactic free to align with the dot. Here the pointer was 3D printed with its tip indicate the geometry focus of the fast transducer. In step 2, the pointer was switched to the transducer, the fast transducer, and then the fast transducer was moved to the target location using its coordinates in reference to the bregma as determined by the mouse brain atlas. The targeting method can be easily adapted by the neuroscience community. After simulation and calibration of the midi-fast transducer, we perform fast BVD in mice using 3 fast transducers at different pressure level, 0.2, 0.4, and 0.57 MPa. Based on MRI imaging at different frequency and pressure, we can find the higher frequency can decrease the BVD opening volume at the same pressure as the 0.4 MPa. I would like to highlight the BVD opening volume as small as 2.4 cubic millimeter was achieved using 6 MHz at 0.4 MPa. Such a small BVD opening volume allows precise drug delivery to the mouse brain. Here we evaluate the targeting accuracy of the midi-fast device by comparing the desired location with the centroid of the actual BVD. We firstly choose a target based on brain atlas as the yellow dots shown. Then we observed the BVD result using MRI after fast procedure. After that, we compare the desired target and the BVD opening centroid as indicated by the blue dots to quantify the offset after the co-registration. We found the offset for the different frequency at different pressure were around 0.6 millimeter. No significant difference was found among the different frequency from 1.5 to 6 MHz. It means our fast device can achieve precise targeting. Cavitation index and mechanical index were proposed before as an indicator to predict the fast PVD drug delivery outcome. Cavitation index is defined as pressure over frequency, and the mechanical index is defined as the pressure over square root of the frequency. We can find the gadolinium value have a strong linear correlation with the CI and MI. And the correlation coefficient of CI is slightly higher than that of MI. Those findings are important as they indicate our fast device can precisely control fast PVD outcome. After performing MRI, we also inject model drug Evans Blue just to test to the mouse to evaluate the drug delivery efficiency of the different transducer at different pressure. Here we show the representative imaging of ex vivo mouse brain slice. The bright color you see represent the delivery Evans Blue. As you can see in the coronal view of the ex vivo brain slice, the drug delivery almost covers the whole brain depth at 1.5 MHz, but it was decreased at the higher frequency to 6 MHz. The drug delivery was well confined with the targeting region. This highlights the unique advantage of higher frequency fast transducer in achieving precise PVD opening. The Evans Blue value also had a strong linear correlation with CI and MI, showing that we can use CI and MI to precisely control the drug delivery outcome. The result is consistent with gadolinium delivery outcome, showing that our fast device can precisely control the drug delivery outcome. In conclusion, we developed a tunable, easy-to-use, and high-precision fast device for fast PVD in mice. And if you are interested in our work, please check out our recent IEEE paper. I would like to thank our whole team, especially my mentor, Dr. Hong Chen. And I also want to thank the funding agency. Thank you everyone to attending my presentation. I'm happy to answer any question.